Hello everyone and welcome to today's session. And today we'll be discussing the theory related to the chapter Human Health and Diseases. Okay. In today's section, we'll be discussing NCERT section 7.3. Okay, so this is the disclaimer. So what are the objectives of today's session? So today we'll be talking about AIDS. Okay. So we will understand what is AIDS, and we will also talk about the causative agent, which is HIV, and what are the mode of transmission of AIDS, which is a sexually transmitted diseases. Okay, we'll also talk about the cancer. Okay, we'll understand what cancer is and how it can be prevented. What are the treatments of cancer? We'll talk about this. Okay, clear? So we are going to talk about the mechanism of HIV infection as well. Okay, this is the retrovirus which causes AIDS. We'll also talk about how this virus affects the immune system. Okay, and in addition to this, we'll be also talking about the stages of AIDS progression. We'll also discuss the social aspects, misconception, and importance of awareness about AIDS in society. Clear? So today we'll be talking about AIDS as well as cancer. Then we will summarize this entire session. We'll discuss some NCERT-based questions, and then there'll be some keynotes. In the end, there'll be some interesting questions that will help you memorize these topics. So let's begin with AIDS first, and then we'll talk about the cancer. Okay, so what is the full form of AIDS? So AIDS is acquired immunodeficiency syndrome. So you can, by seeing the name, so what you are understanding here. So now notice how the name itself tells us a lot. Acquired means it is picked up during a person's life, not something you are born with. Immunodeficiency points to a problem with the immune system, and the syndrome means it is a collection of symptoms. Okay, how can you define AIDS? It's a disease where the immune system becomes weak, but this weakness is not present from birth. It's acquired later in the life. Okay, so in AIDS, what is happening? The person is acquiring this disease during its lifetime. Okay, so this AIDS, this is different from the genetic diseases. This is not a congenital disease. Congenital disease, these are diseases which are from the birth. Okay, so this AIDS, this is not a congenital disease. It is acquired during the lifetime. Now, what is the causative agent of AIDS? So it is HIV, which is human immunodeficiency virus. So this virus attacks and weakens the immune system, making the body vulnerable to other diseases. Now let's look at the type of the virus HIV is. So HIV is a retrovirus, means its genetic material is RNA, not DNA. And this RNA is wrapped in a protein coat. Now, AIDS was first reported in 1981. Now, this is relatively recent in medical history. Now, since then, it has become a major global health issue affecting millions of people worldwide. Now, here you can see this image. This is the image related to AIDS. This is a symbol, okay, for fighting against AIDS. Now, let's talk about the transmission of the HIV infection. How HIV is transmitted from one person to another person. So first, HIV can be transmitted through sexual contact with an infected person. Here. So, during the sexual intercourse, the person can get AIDS. It can be transferred from the infected person to the healthy person. So that is why safe sex practices are so important in preventing HIV. Another way HIV spread is through transfusion of contaminated blood and blood products. Now, HIV can also be transmitted by sharing infected needles, which is common among people who use intravenous drug. Now, the virus can also move from an infected mother to her child through the placenta during pregnancy. So, placenta is like a bridge between mother and the fetus. If the mother has HIV, the virus can cross this bridge, which is placenta, and infect fetus as well. This is why prenatal care and testing are so important for expecting mothers. Here you can see these are the modes of transmission of these sexually transmitted diseases such as AIDS. So it can be transferred through or during the unprotected intercourse, by needle reuse, during blood transfusion, and as well as during the pregnancy if the mother is having the HIV. Now there is one important note here about the AIDS transmission. So notice that HIV is not spread by simple touch or physical contact. This means you cannot get HIV from hugging, shaking hands, or sitting next to someone who is having HIV. It is very important to clear up this myth as it helps reduce unnecessary fear and stigma around people living with HIV. Now let's have a look at how HIV actually spreads. We have already talked about it. So HIV is transmitted only through certain body fluids like blood, semen, vaginal fluid, and breast milk as well. Everyday activities like sharing food, using the same toilet, or casual contact do not spread HIV. Only direct contact with these specific fluids can transmit the viruses. Here, finally, it's important to understand that after someone is infected with HIV, there is a time lag before symptoms of AIDS appear. The symptoms of the AIDS, they appear after some years, maybe 5 to 10 years. So this period can range from a few months to as long as 5 to 10 years as well. So you can see the symptoms are appearing quite late. So this delay is why regular testing and early detection are so important. Even if someone feels healthy, then also regular testing and screening of this STDs, it is very important. So this will prevent the spread of these diseases. Now let's talk about HIV replication cycle. So HIV, this is the virus, this is the retrovirus. It has RNA as the genetic material. Now, what is the mechanism of action? Let's talk about how HIV infects a person, infects a host. So let's talk about the entry. So after HIV enters the body, it does not attack just any cell. It specifically targets macrophages, which are like the body's security guards. Then what happens? There is a process known as reverse transcriptase, reverse transcription, in which an enzyme reverse transcriptase is involved. So HIV has RNA as the genetic material, but the host cell, such as human, the human cell contains DNA as the genetic material. So this virus uses a special enzyme called reverse transcriptase to convert its RNA into DNA. And this process is known as reverse transcription. Now, next step is the integration. Now, the viral DNA gets integrated into the host cell DNA. So, this step is crucial because it makes the infection long-lasting and hard to eliminate. Now, you can see here the viral DNA, the viral genome is now integrated into the host genome. And that's why it is very hard to eliminate. We're talking about the mechanism of action of HIV. Now, You have to notice here that the macrophage acts like an HIV factory. Macrophages are usually our body's defenders, like security guards, but HIV turns them into virus-producing machines. So this is how the virus keep multiplying and spreading through the body. So they are multiplying within the macrophages. At the same time, HIV also attacks the helper T lymphocytes, also known as the T helper cells, which are like the generals of our immune army. Okay, they also provide immunity. Now the virus enters these cells, make many copies of itself and release new viruses. So this weakens our immune system, making it harder for the body to fight off the infections. Okay, so now the cells, they are bursting, macrophages and T helper cells. Okay, both of these cells, they are dying, so their number will reduce. So our immunity, our immune system weakens because of this. Now here in this image, you can see an image that summarizes the HIV replication cycle. You can notice how the virus enters here, uses the cell machinery to make more viruses and then releases them to infect other cells. So this cycle is what makes HIV such a persistent and challenging virus to control. So here you can see in this image, this is a retrovirus, which is HIV. It has RNA as a genetic material. Okay. So any retrovirus, okay, this mechanism will be followed by any kind of retrovirus such as HIV. So here you can see it is integrating its genetic material into the host cell. Here the host cell is macrophage. Then by the process of reverse transcription, RNA is converted into DNA. This DNA gets integrated into the host genome. And then it is using the host machinery to produce more copies of itself. Okay, so viral, you can see here, new viruses are produced here. Here, and before that, new viral RNA is produced. By using host machinery, around this viral RNA, protein coat is assembled, and this is how new viruses are formed. These viruses will be released, okay, and then they can infect the other cells as well. Now finally there is an immune collapse. Now let's have a look at what happens when the virus multiplies. Now the new viruses are released from an infected cell and go on to attack other helper T cells. Okay, so these cells are now infecting the helper T cells. So this is how this infection spreads within the immune system. As the cycle repeats, the number of helper T lymphocytes keep dropping. Now these cells are like, you can say, coordinators of an immune response. They provide protection against the invaders. Now, over time, the immune system becomes so weak that even common infections can become life-threatening. So this is why immune collapse is so dangerous. Why is immune collapse happening? Because the reduction in the number of T helper lymphocytes. Here again, you can see in this image, is an image showing you HIV replication cycle. Okay, let's talk about the symptom of AIDS. First, notice that people with AIDS start suffering from infections that a healthy person would usually overcome easily. The infection can be caused by bacteria like mycobacterium, as well as viruses, fungi, and even parasites such as toxoplasma. Now, as the disease progresses, the patient becomes immunodeficient, meaning their immune system is too weak to protect them. This is why people with AIDS are much more vulnerable to infections. The common symptoms you might see include repeated bouts of fever, ongoing diarrhea, and noticeable weight loss. These are the symptoms of AIDS. These are the symptoms that the body is struggling to cope with infections and cannot maintain normal health. Okay, let's talk about the diagnosis of AIDS. Now, one of the most common tests used to diagnose AIDS is called ELISA, which stands for Enzyme Linked Immunosorbent Assay. So this test is widely used because it's sensitive and can screen large number of people efficiently or large number of samples efficiently. Now let's move on to the treatment. It's important to know that while we have ways to manage AIDS, there is currently no complete cure for AIDS. Okay, usually the STDs caused by viruses, they are very difficult to treat, and there is no cure for STDs caused by virus. Bacterial, okay, STDs can be easily cured. Now let's move on. Now the main approach for treating AIDS is with antiretroviral drugs. These medicines work by slowing down the multiplication of the HIV virus in the body. However, they can't completely stop the virus or remove it from the body. Even with antiretroviral drugs, the treatment can only extend the patient's life and improve its quality for some time. Unfortunately, these drugs cannot prevent the eventual outcome as the disease is still fatal. Okay, so there is no cure for AIDS. Only the symptoms can be managed by antiretroviral drugs. Okay, how can these STDs can be prevented? Okay, how can AIDS can be prevented? Now, the slogan "Don't die of ignorance" emphasizes that many infections are preventable through knowledge and conscious behavior. The slogan "Don't die of ignorance" emphasizes that many infections are preventable through knowledge and conscious behavior. Let's talk about some key strategies for preventing AIDS. First, spreading awareness and educating people about AIDS is like putting a warning signs before a dangerous curve in the road. Now, next is ensuring the use of disposable needles and syringes. This is fatal. Clear? To think of it like using a new toothbrush instead of sharing one. Reusing needles can transfer the virus from one person to another person. Now, screening blood before transfusion is another key method. Okay, this is another key measure. Okay, here the screening of the blood before transfusion should also be done to prevent the spread of these sexually transmitted diseases because they are spread through blood as well. Promoting safe sex practices is also crucial. Yeah. So education should be given, awareness should be spread regarding safe sexual practices and how to prevent the sexually transmitted diseases. Organizations like NACO, which is National AIDS Control Organization, and various NGOs play a big role in spreading information and supporting prevention efforts. Okay, now let's summarize this topic. We talked about AIDS today. Okay. So we have discussed that AIDS stands for acquired immunodeficiency syndrome. It's caused by HIV, which is a retrovirus. HIV was first reported in 1981. Okay. We have also talked how AIDS or HIV spreads. The main way are through sexual contact, sharing contaminated blood or needles, and from mother to child during birth or breastfeeding. Now, it's important to highlight that these are direct ways the virus can enter the body. So, understanding these routes help us to know how to protect ourselves. So, HIV cannot be spread by casual touch, like hugging or natural or shaking hands. It only spread through certain body fluids. Also, symptoms of HIV infection might not show up for months or even years. This is why some people may realize they are infected right away. Okay, only some people they come to know that they are infected. because the symptoms, they appear very late. Means the infection is there, but the symptoms are not appearing. This is known as asymptomatic. Clear? Now, we also talked about the HIV replication cycle. So HIV, they enter into special kind of immune cells called macrophages. Now, this virus uses an enzyme reverse transcriptase to convert RNA into DNA. Then DNA is integrated into the host cells, and then this virus is using the host machinery to produce more of its own kind. Okay. After that, these viruses, they will infect the helper T lymphocytes, progressively weakening the immune system. So the number of T helper cells, they will keep on reducing. They will keep on declining, thus weakening the immune system. Now, symptoms of AIDS include vulnerability to infections, fevers, diarrhea, as well as weight loss. Now, ELISA is a very, you can say, commonly used for the diagnosis of AIDS. So this test checks for antibodies against HIV in the blood. It's a simple laboratory technique that helps identify if someone has been exposed to virus. Now, antiretroviral drugs can extend life but cannot cure AIDS. So there is no cure for AIDS. Only AIDS can be managed through antiretroviral drugs. Clear? Now, prevention focuses on awareness, safe sexual practices, clean needles, and blood screening as well. Okay. So awareness should be raised regarding safe sexual practices. Clear? And these AIDS or these STDs, they can also be prevented by using contraceptives such as condoms. Okay. Now you can see certain organizations like NACO and various NGOs play a big role in educating people about AIDS, preventing and controlling the spread of these STDs. So their work helps communities understand the risk and how to protect themselves. Okay, so this was all about AIDS. Now let's discuss some NCERT-based question related to this topic. So first question is, explain the molecular mechanism by which HIV transforms its RNA genome into DNA and why this makes it unique among viruses. Let's have a look at the answer. Okay, HIV is a retrovirus that uses reverse transcript enzyme to convert its RNA genome into DNA, which then integrates into host cells' DNA. Now, this process of genome replication is unique as viruses carry DNA genomes or use RNA without DNA conversion into RNA. Okay. So, why is this unique? Because most viruses, they have DNA as the genetic material, or they use RNA without converting it into DNA. Here, HIV, what is HIV doing? It has RNA as a genetic material. It is using this enzyme reverse transcriptase to convert RNA into DNA first, because the host cell, it has DNA as the genetic material. Now the viral DNA, okay, HIV viral DNA, it will get integrated into the host cell's DNA after the process of reverse transcription. Next question, in the context of AIDS pathogenesis, why does the destruction of helper T lymphocyte have such devastating consequences for immune system? So let's have a look at the answer. Okay. Now, helper T lymphocyte play a central role in coordinating the immune responses. So these are the cells which provide immunity. Now, if these cells are destroyed or their number decline, then this will weaken the entire immune system. Okay, leaving the body vulnerable to opportunistic infections as well as cancer as well. This is why people with AIDS often get sick from diseases that wouldn't usually be a problem. Okay, now key notes related to AIDS. So AIDS is caused by HIV, which is a retrovirus, and AIDS is transmitted through body fluids. Okay, during sexual intercourse, this AIDS can also spread by blood or during blood transfusion, so using infected needles, and from mother to child as well. Now, HIV actually affects our body. HIV specifically targets the helper T lymphocytes, and you can see as the number of helper T lymphocytes decrease, the body is vulnerable to the opportunistic pathogens, opportunistic infections. And this HIV virus, it uses reverse transcript enzyme to convert its RNA into DNA, and then the viral DNA gets integrated into host cells DNA. Symptoms include weakened immunity, fever, diarrhea, weight loss. Clear? And symptoms, they might appear after months or even after years after infection. Now, diagnosis through ELISA test. So, ELISA can be used for detection of the AIDS. Okay, this is a test for the diagnosis of the AIDS. And antiretroviral drugs can be used to manage the symptoms of the AIDS disease. Prevention of AIDS can be done through raising awareness, safe sexual practices, using clean needles, blood screening, and there are certain organizations like NACO, okay, National AIDS Control Organization, and NGOs that you can say spread awareness regarding this issue. Okay, clear? So this was about AIDS. Now let's talk about the topic cancer. Okay, let's discuss cancer now. So cancer is one of the most feared diseases, and it's mainly defined by the abnormal and uncontrolled division of the cells. Okay. Now, the key feature of healthy normal cell is that they will stop dividing and growing when they touch each other. And this process is known as contact inhibition. But these cells, okay, they lose the property of contact inhibition and they keep on dividing. Now, cancer cells appear to have lost this property. Which property? Contact inhibition. That's why they keep on dividing. Now, as a result of losing this ability of contact inhibition, cancer cells continue to grow and pile up on top of one another, forming masses known as tumors. Here you can see a diagram showing the difference between normal cells and cancer cells. Notice how the normal cells are organized and stop growing when they touch each other, while the cancer cells keep piling up, forming a tumor. Let's talk about the type of cancer. Okay, so there are mainly two main type of cancer. Okay, benign tumors and malignant tumors. The main difference between benign and malignant tumors is their behavior. Benign tumors are slow growing. They stay in one place and do not spread to other body parts. Whereas malignant tumors, which are cancerous, they grow fast, invade nearly healthy tissues, and can spread through the blood to other areas of body to form new tumors. Now, this ability of malignant tumors to spread is known as metastasis. And this is what makes them so dangerous and often life-threatening. Here, unlike benign tumors, which are generally not harmful unless their size causes problems by putting pressure on the vital organs. Okay, here you can see this is an image showing you benign and malignant tumor. Malignant tumor, okay, this is more fatal. Okay, this is more dangerous as compared to the benign tumor. This you can see the benign tumor is spreading from one area to another area. What are the causes of cancer? Okay, let's talk about the physical carcinogens. So what does physical carcinogens include? So here you can see this table is talking about the physical carcinogens. So here you can see ionizing radiations and non-ionizing radiations. This can cause cancer. The difference between ionizing radiation and non-ionizing radiation is also shown here. So ionizing radiations are like X-rays and gamma rays, and they are very powerful. They are fast-moving waves with a lot of energy and a shorter wavelength. And this allows them to penetrate deep into your body and directly damage the DNA. And this can also cause mutations and possible cancers. Exposure to high levels can cause burns and radiation sickness as well. Now, non-ionizing radiation like UV rays from the sun or tanning beds have less energy and longer wavelengths. So they mainly affect the surface like the skin, causing damage that can also lead to DNA mutations and increase the risk of skin cancer. So while UV rays don't penetrate as deeply, prolonged exposure to high doses can still have harmful effects. So you can see these are physical carcinogens, ionizing radiations and non-ionizing radiations. Both of them can cause cancer. Now let's move on. Let's talk about the chemical carcinogens. So these are substances that can trigger cancer by causing mutations in the DNA. Now tobacco smoke is a prime example of chemical carcinogen. It's a leading cause of lung cancer worldwide. Other chemicals found in pollutants, food additive, etc. These can also cause cancer. Here you can see in this image, you can see tobacco. It can affect the lungs. Okay, tobacco smoke. This is a major cause of lung cancer. So here you can see on the left they are showing the damaged lung and they are also showing a normal lung here. Next one are the biological carcinogens. So these biological carcinogens include viruses as well as bacteria and some parasites as well, and which can disrupt normal cell processes. Now here you can see this table is showing you comparison of viral oncogenes and cellular oncogenes. Okay, viral oncogenes are written as V-onc and cellular oncogenes are written as C-onc. They are also known as proto-oncogenes. So let's understand the difference between the viral oncogenes and the cellular oncogenes. Okay. So you can see here biological carcinogens like certain viruses can cause cancer by messing with the genes that control cell growth. Here you can see viral oncogenes. What is the origin? They are found in cancer-causing viruses. And cellular oncogenes, they are found in the normal cells. Now, what is the normal function of viral oncogenes? They promote viral replications and cell transformation. Whereas cellular oncogenes, they regulate normal cell growth and division. Now, these viral oncogenes, they are constitutively active. Means they are always active. Constitutively means they are active all the time. And you can see cellular oncogenes, they are usually inactive, but they require activation. If they get activated, then they can cause cancer. Okay, so this is the difference between viral oncogene and cellular oncogene. Okay, here you can see in this image showing you proto-oncogene and the oncogene. Clear? So you can see here in this image how these oncogenes, they are activated and they can cause cancer. Okay, this is clear. Now proto-oncogenes, they get activated and they can cause cancer. Usually they are inactive, but due to certain change or due to certain stimulus, they get activated and they can cause cancer. Okay. Now some more differences. Here, if we talk about viral oncogenes, it has no cellular regulation, and in the case of cellular oncogenes, they are tightly regulated by cellular mechanism. Now, these viral oncogenes can directly cause cancer without introduced into cells, and the cellular oncogenes, they can cause cancer when abnormally activated. Now, discovery, they were first discovered in viruses, and these oncogenes, cellular oncogenes or proto-oncogenes, they were later found to be derived from the cellular genes. Okay, this is clear. So basically, proto-oncogene, this is a normal gene that promotes cell growth and division. This proto-oncogene can become an oncogene if it gets mutated or it gets altered, and this can lead to uncontrolled cell proliferation that can cause cancer. Okay, so these proto-oncogenes are actually normal genes. Okay, clear? But they can cause cancer. Now, how can you detect cancer? So by biopsy and histopathological studies, we can detect cancer. Now, in this process, a small piece of suspected tissue is cut into thin sections, stained and examined under a microscope, and this allows doctor to see the shape and arrangement of the cells, helping them identify if the tissue is cancerous or not. Okay, let's discuss about the blood and the bone marrow tests. So these are especially important for disease like leukemia, which affects the blood cells. Okay, and doctor looks for increased cell counts in the case of leukemias, which is a type of blood cancer. Too many abnormal cells in the blood or bone marrow can be a clear sign of disease. Radiography, okay, which include X-rays, and CT, which is computed tomography, and MRI, magnetic resonance imaging. So these are non-invasive ways to see inside the body. So these techniques are used to detect cancers of the internal organs. For example, CT scan can reveal tumors in the liver or lungs that can't be felt from outside. Okay, CT uses X-rays to generate 3D images, giving doctors a detailed look at the body structures. MRI, on the other hand, use strong magnetic fields to create images, which is especially useful for soft tissues like brain. Another detection method involves antibodies. So these are special proteins made by the immune system that can recognize and bind to specific targets. Now, doctors use antibodies against cancer-specific antigens. These antigens are like unique ID tags found only on cancer cells. So the antibodies help highlight or even capture these cells for detection. Now, let's talk about the treatment of cancer. Okay. Now, first treatment is surgery. So surgery involves physically removing the tumor from the body, much like cutting out a damaged part from a machine to keep it running smoothly. The main goal of surgery is to remove as much as the tumor mass as possible. So this helps prevent the cancer from spreading and can sometimes cure the patient if all cancerous tissue is removed. Next, we have radiotherapy, also called radiation therapy. This method uses high energy rays to target and destroy cancer cells. During radiotherapy, doctors carefully aim the radiation at the tumor or kill cancer cells while protecting the normal tissues around it. So this precision is crucial to minimize side effects and ensure healthy cells can recover. Okay, next is chemotherapy. So chemotherapy is a powerful type of medicine used to fight and kill fast-growing cancer cells. Now, while this medicine is very effective at killing cancer, it can also affect some of the body's healthy cells as well. Okay, such as hair follicles and the digestive system, which is why patient can experience side effects like hair loss and nausea. This chemotherapy, often combined with the combination of other treatment methods like surgery or radiotherapy, is very effective in the treatment of cancer. Next method is immunotherapy. It works by stimulating the immune system to better recognize and attack abnormal cells like cancer. Now, immunotherapy as a treatment that helps your body own immune system fight cancer. Okay, so here the immune system is activated so that you can fight cancers. So here patients are given biological response modifiers such as alpha interferons, which activate their immune system and help in destroying the tumor. Let's summarize this topic, cancer. So we've discussed cancer. It is a disease where cells begin to divide uncontrollably. There are two main types of cancer, benign and malignant cancer. Benign tumors grow slowly, they stay in one place like a harmless lump, while malignant tumors grow quickly and can spread to other parts of the body through a process called metastasis. We also discussed about the causes of cancer. So they are caused by three kinds of carcinogens: physical, chemical, and biological carcinogens. You should remember the examples of each one of them. Then we also talk about the difference between the normal and the cancerous cells. The key difference is that the cancer cells, they lose contact inhibition. Okay, while the normal cells, they retain the ability of contact inhibition. After that, we have discussed how cancer is detected. So doctor use methods like biopsy. Okay, where a small tissue sample is examined, blood tests to check for abnormal markers, and imaging techniques such as X-ray, CT scans, and MRI to visualize tumors inside the body. Now, proto-oncogenes are like the body's growth switches. So basically, proto-oncogenes, they regulate the normal cell growth and division. These proto-oncogenes can get activated, or they can get altered or mutated, and then they become oncogenic. Means they can cause cancer then. So if these proto-oncogenes, if they are abnormally activated, they can turn into oncogenes and cause uncontrollable growth leading to cancer. A treatment for cancer can be remembered by the acronym SCRI, okay, which means surgery to remove tumors, chemotherapy using drugs to kill cancer cells, radiotherapy using radiation, and immunotherapy, which helps the immune system fight cancer. Let's discuss some NCERT-based questions based on this topic, cancer. So question is, what is the fundamental difference between normal cells and cancer cells regarding contact inhibition? So let's have a look at the answer. See, normal cells stop dividing when they come in contact with other cells. This is known as contact inhibition. Whereas cancer cells, they have lost this property and continue to divide uncontrollably. Now next question, compare viral oncogenes and cellular oncogenes. So let's have a look at the solution. So here you can see this is the difference between viral oncogenes and cellular oncogenes. So viral oncogenes, they are derived from cellular genes captured by the viruses, whereas cellular oncogenes, these are, okay, the natural components of the cellular genome. Then viral oncogenes, they are modified, they are the mutated versions. And then cellular oncogenes, they are the normal cellular gene. Okay, clear? Actually, the proto-oncogenes, they get activated and they can turn into oncogenes, which causes cancer. Now let's talk about the regulation. Viral oncogenes are poorly regulated or constitutively active. Now cellular oncogenes, they are well regulated under normal conditions. Here, under normal condition, it is saying it is well regulated, but it can be abnormally activated and then they become oncogenic. Now, viral oncogenes, they are always active, and cellular oncogenes, they can be turned on or off as well. Now, natural function of viral oncogenes promotes viral replication and cell transformation. What is the natural function of cellular oncogenes? Usually they control the normal cell growth and division. So proto-oncogenes, they control normal cell growth and division, but if they get abnormally activated, they can also cause cancer. Now, viral oncogenes, they are directly oncogenic, whereas cellular oncogenes, they are only oncogenic when mutated or when altered or when abnormally activated. Let's discuss some keynotes. There were two main types of tumors, benign and malignant tumors. Okay, benign tumors, they are localized and slow growing, and malignant tumors, they are rapidly growing and metastatic. Okay, now they are caused by three kinds of carcinogens: physical, chemical, and biological. Detection method include biopsy, blood test, radiography, and antibody test. Okay, Now there are two kind of oncogenes, viral oncogenes and cellular oncogenes. We have talked about the difference between them as well. Now, detection of the cancer can be done through these techniques. We have already discussed them. Now, this chemotherapy has a lot of side effects. So, chemotherapy can be combined with radiotherapy, immunotherapy, okay, and this will give better results. Clear? Now, there are some interesting questions. So, these questions are: cancer cell loss of contact inhibition is both their strength and weakness. You have to critically analyze this statement in context of cancer treatment strategies. The next question: compare and contrast the mechanism of physical and biological carcinogens, and how does this understanding influence prevention strategies? You have to critically analyze these questions. Okay, so with this we end this session. Okay, so this is the end of the theory. Here, I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.